So it's really a great pleasure to be here. I apologize for not even attempting to speak in French because it's a very long time since I learned it at school in Scotland. So it's nice to be in Europe, but please indulge me in English here. So let's begin. So what I'm going to talk to you about today is really just one story of work in the lab, which is dealing with the localization of kinases in pathological situations. And it really begins by looking at the inside of a cell. And this is a wonderful drawing that was done by David Goodsall at Scripps Institute in La Jolla, which is an artist's impression of the inside of a cell. And it's very accurate. It's a 2 million-fold magnification of an electron micrograph. It's incredibly congested. Each of these blobs is a structured protein. And it's the spaces between these that are really important for all the signals that we're talking about and the movement of information from one side of the cell to the other. Obviously, one of the ways that this happens is phosphorylation. So localization of kinases and phosphatases, or deubiquitinases and ubiquitin ligases, or acetylases and deacetylases, is very important to use the information to move it from one part of the cell to another. Now, what sort of my career has been focused on has been understanding the intracellular organization of these signaling complexes, or as we now call them, signaling islands, because they're really, as you'll see, quite small. And this has resulted in the discovery of a family of proteins called A-kinase anchoring proteins, or ACAPs, that are distributed throughout the cell. So when we began, I think there was one ACAP gene known. And currently in the human genome, there's over 60 ACAP genes. And I'm sure there will be more that will be discovered. So there are a convergent gene family that target kinases and phosphatases and signaling molecules to unique locations within the cell. And this is just a few examples of some of the biological roles that they play. I think really importantly to understand these, you really need to begin with the structure. And this is a cryo-EM structure of the simplest ACAP complex that we could find. The ACAP is in gold. It's called ACAP 18. They have very unimaginative names. It's bound to this structure, the docking dimerization domain, which is a region of the regulatory subunit of the cyclic AMP-dependent protein kinase that folds into this sort of helical bundle that binds with very high affinity to a helix on the ACAP. And that's the anchor. So this is the anchoring part. Next to this are these regions of sort of indiscriminate, intrinsically disordered regions, which are very, very important. And hanging off those are the two cyclic AMP binding pockets of the regulatory subunit. And the kinase, the part that does the phosphorylation, is in blue, is in the center. So by going through the sort of anatomy of this complex, I think it's really important to understand that each part plays a role in the biology. And this structure, we solved this part. It's very important to recognize that this work was done by Susan Taylor's lab in San Diego and many other people. So this has been a real collaborative effort to get to this model. So what does it do? Well, one of the first and important things is when you target something to a particular location, two things happen. One, you can focus the signal, and secondly, you can insulate the signal so that other parts of the cell don't respond when you activate this particular probe. And for that to happen, this has to remain in contact. And one of the things that we discovered a few years ago was, contrary to all of the biochemistry that I learned and was actually trained by the people that discovered this enzyme, Ed Krebs and Eddie Fisher, this complex, although it's separate subunits, remains intact inside cells. And so that intrinsic disorder region I talked about is really important to give motion to the complex and reach so you can unleash the phosphorylation events. This is maybe better shown here, where you can look at these cryo-electron microscope gaps. I'll show you here. So here we're starting with a class average for their nearest neighbors of about 1,000 particles. And you see this very tight triangular complex. You can begin to see how that looks like the real structure here. Just either higher or lower resolution, depending on how you want to look at it. As I move along here, if I can do this, you'll notice that these structures change in their orientation so that you go to this extended structure as shown down here. So that motion and that movement is really important to be able to reach different substrates in different parts of the cell. And with the beauty of electron microscopy and the sort of precision of structural biology, we can actually calculate these distances. And it's somewhere between 2 to 4 angstroms. Now, I've redrawn this on the back of David Goodsall's diagram. And what you can see is that that range of motion is between 2 to 4 molecules. So these nanodomains are extremely small. They're in 3 dimensions, and it's within these confines that the signaling happens. So again, if I go back to this idea of focusing signals and insulating signals, you can see it in this diagram. And so that's really what I'm going to talk about for the next however long I have here. So when we figured this out, and there was a lot of us that were working on it, we realized that this tight location has profound consequences for function. When you think of a broad specificity enzyme such as protein kinase A that has thousands of substrates, in the confines of these spaces, there's very few substrates. So localization by ACAPs determines the substrate selectivity of this multifunctional enzyme that uses common second messengers and G proteins and GPCRs to be activated, but gives very specific responses in response to individual hormones. Another sort of logical extension of that is the idea that if this is the way that things work, mutations or changes in these complexes are going to have pathological effects. And it's really that I'm going to talk about today. Our lab works on 4 or 5 different models at the moment, but what I'm going to talk about today particularly is non-ACTH or adrenal Cushing syndrome. Now, this is something that many of the people in the audience know a lot more about than I do. And it's really, we've discovered that disruption of PKA anchoring underlies aspects of Cushing syndrome. By no means all of it, but certainly aspects. So for those of you that don't know, Cushing syndrome is a disease of the overproduction of cortisol, the stress hormone cortisol. It has some fairly unpleasant side effects, weight gain, changes in the adrenal glands. It's very common in dogs. In fact, I'm now at the point where I can walk about my neighborhood and identify dogs with Cushing syndrome because they're quite distinct. What's probably the most distressing is some of the comorbidities, which are somewhat reminiscent of type 2 diabetes. You've got hypertension, high blood sugar, heart disease, digestive problems. There's also anxiety and depression. So this is a relatively rare endocrine disorder, but a fairly unpleasant one. And generally, treatment is either removal of parts of the adrenal gland, or people just have to have it corrected by various different ways. So what we became interested in is some work that was done here and in other labs throughout the world about 7 or 8 years ago, identifying point mutations in the catalytic subunit of protein kinase A that lie on that interface between the regulator and the catalytic subunits. The classic mutation that occurs in about 45% of patients, or somewhere in that range, is this leucine to arginine 205 mutation. We also identified another mutation, which is a tryptophan 196 to arginine. This is less common, but you'll see that when we looked at these 2 different point mutations, they had very different effects in the signaling environment. So with that in mind, what we really began with was we reckoned that both of these mutations would abolish the ability to bind the regulatory subunits of PKA, and hence release or not allow these Cushing's mutants to be incorporated into ACAP signaling islands. And so from a sort of very simple biochemical perspective, we immunoprecipitated the ACAP in cells that expressed each of these mutants in wild type and confirmed that, yes, they don't bind the regulatory subunits and they're free to move around the cell, which is not what normally happens. This is just reciprocal IP. And so the next thing was, if you've changed the location of the kinase, have you changed its substrate selectivity? Because it has access to different substrates. And so to do this, we took advantage of this quite sophisticated mini turbo proximity proteomics, where you attach a biotin ligase to the kinase. It's either incorporated or not into the ACAP signaling complex. There's a range of about, I think it's 5 to 10 microns that you can then get biotinylation. You identify the peptides, and you can determine the kind of proteome and the phosphoproteome that surrounds this area. So we've done this for both of these mutations. I'm just going to show you the data for the W196R mutation here. And what you can see in this volcano plot, or I hope you can see, in pink are the signals that are enriched. So these are more preferentially targeted in this configuration. And here are the signals that are depleted. And this is the first interesting thing that we discovered. Many of the depleted signals, whether it was phospho proximity proteomics or just regular proximity proteomics, as I'm showing here, turned out to be the ACAPs. So that fit the model that if you didn't get this incorporated into the complex, you were going to have some pathological events. So could we see this in cells? And so this is where we took advantage of live cell imaging using super resolution imaging and photoactivatable RFP. So when you shine 4 or 5 nanometers of light with your laser, at a very particular point, you can activate this RFP signal. Hope you can see this on the membrane here. This is the normal configuration with all the wild type proteins. Nothing happens because nothing moves. We've actually challenged these cells with isopaternal, so this complex remains intact. Here's the same experiment with the L205R Cushing's mutant. What you see is very rapidly that molecule moves. It's excluded from the nucleus. You can see the nucleus here, and this is over a very rapid time frame. And you can calculate all this. You can quantify. It's shown again here in sort of a sort of time frame, just to really emphasize the point. This is the important data. It's the quantification for many cells. We also did the same thing looking at a different location using a different ACAP. In this case, we're looking at DACAP1, which is at the mitochondria. This anchoring protein called ACAP79 is at the plasma membrane. You see the same result. So non-anchoring has significant effects, and it's associated with a single point mutation. Here's the quantified data in adrenal cells, which is basically the same stories I've just shown you. There's subtle differences between L205R and W196R, which I'll get into a bit later. So these variants are excluded from ACAP signaling islets. So that means that they're free to roam around the cell, indiscriminately phosphorylating things, or very specifically phosphorylating the wrong thing. We don't really know the answer to that yet. But that's all well and good in a cell. What about in patient tissue? So these are actual sections of adrenal gland. This is normal adjacent tissue. The green signal is the location of PKAC subunit. It's highly organized in the cell. From the same sample, we looked at an adenoma, and you see a very, very different pattern, a much more diffuse location. I think we'll be able to see this a bit better when I actually expand these, where you can really compare the native situation to the disease. It's quite dramatic. And we can actually see this. We've now looked at, I think, maybe 30 different tissue sections. So we observed this in multiple cases. And this is, in this particular case, this is a W196R mutation. I'll show you an L205R mutation later. So what's the difference? It's a single point mutation. In fact, these two mutations I'm talking about both have the same residue mutated. It's arginine. It's 9 amino acids apart. That's going to be important later. They're excluded from ACAP signaling islets. But here's the other interesting thing. When we do the biochemistry and do native mass spectrometry on these individual mutants, they're quite different. There's a point mutation that's different in the amino acid sequence, but the covalent modification of each of these forms is dramatically different. There are 11 moles of phosphate incorporated into the wild-type catalytic subunit that's necessary for its activity, about 6 moles in the W196R, and maybe 1 mole of phosphate in the L205R. This translates, at least in vitro, to changes in activity. These two forms have essentially normal activity. This is actually dose response curves using chemtide as a substrate. The L205R mutation is about 20 to 30-fold lower in its activity. I'll say that that is surprising, and it's also possibly not quite the whole part of the story, because it may be that we're using the wrong substrate here, and that in fact the mutation in L205R changes the substrate selectivity for a different subset of phosphosites. We have some answers to that, but we don't have the full answer to that. But I think it's important to keep that in mind. But nonetheless, when you actually look at these particular mutations in cells, you can see quite a dramatic difference. So here we've added this ACAP79 at the plasma membrane, and then tagged versions of the mutants. The middle lane here, in fact, I think I'll need to walk over to see it myself. You can see a very different intracellular distribution for these mutants. This is the composite image here. So you can see here that there's very different intracellular locations, and this correlates with overproduction of cortisol where we're doing radioamino acid. The surprising thing is that the W196R mutation produces a lot more cortisol than the more common Cushing's mutant L205R, and that holds up quite strongly. So one of the experiments we wanted to do was try and do a sort of molecular biology gene therapy rescue experiment in an adrenal cell by fusing these mutants to the regulatory subunit. We did this with a linker that then allows them to be reincorporated into the ACAP signaling island, as you can see here, where we've actually done this experiment. And then we remeasured cortisol levels. And to our surprise and to somewhat delight, it brought the cortisol levels back down to baseline. So it provides at least the idea that targeting to the right location is very important to maintain normal homeostasis within the cells. And mislocalization is the pathological event. So embedding these mutants within these signaling islands restores normal cortisol release. The other thing that we were really fascinated by was this difference in the signal, because this is the classic Cushing's mutation. That's the most common one. People get sick with it. This is a much rarer mutation. It does happen, but it's rarer. And there's a lot more cortisol produced. So what we decided to do was make a mouse model. And so we were able to make a knock-in. Remember, this is a heterozygous mutation, an adrenal-specific knock-in, so that we had these heterozygotes with a single allele of this W196R and a normal allele, C alpha. This is just proof that we were able to do this. This is work that Mitch Omar, the postdoc that's done all of this work, worked with Stanley McKnight in my department at the University of Washington, that's developed a lot of mouse models for PKD. So the first thing that we found when we looked at the animals, the female body weight was different. Now, for those of you that know a lot about Cushing syndrome is much more common in females than males, and it's accompanied with changes in body weight. And there's about a 20% difference in the body weight of these animals. We didn't see that with the male mice at all. We just saw it with the female mice. Both genders showed differences in adrenal size. So we saw adrenal hyperplasia. These are the adrenal glands. You can see that we can measure them. We can do total protein. We can just actually look at them. And then a little bit of endocrinology. So this is non-ACTH dependent Cushing syndrome, because there's a negative feedback loop when you produce a lot of cortisol that shuts down ACTH production. So if you do endocrine measurements in these animals, you'd expect to see high cortisol and low ACTH, which is exactly what we see. So this was a fairly nice mouse model for Cushing syndrome. And it allowed us to then sort of delve more mechanistically into what we thought was going on. So obviously, when you have hyperplasia, one thing of thinking about is enhanced proliferation. For those of you that know a lot about the sort of interplay between protein kinase A and ERK signaling, it's very complicated and it's context dependent. In this case, PKA activation turns on the ERK signaling pathway. In other cells, it actually does the opposite, turns them off. So we believe that ERK signaling would be upregulated in these cells. And we could actually see this using a phospho-ERK antibody as an index of the activation of the terminal kinase in the complex ERK. You can see that here. And we could also see it in patient samples. So these are tissue samples. It's less clear, but we could still see the same phenomena. This is a quantification here. So we were pretty happy with this. So we've got mislocalization of this mutant allele, and it promotes this mitogenic signaling in Cushing's adenomas. While we were doing this, we also did some additional controls. And one of the controls that was done was to look at another signaling pathway that's involved in cell development and growth, which is the YAP-TAZ hipposignaling pathway. What Mitch noticed was that in cells where ERK signaling was elevated, the YAP-TAZ component of this pathway was depleted. And you can see this in just two examples here, but we saw this in all the mice in both genders, which was surprising to us because we didn't really expect that. This is just how this pathway works. It involves PK phosphorylation. It's a negative phosphorylation in this case that basically prevents 14-3 or recruits 14-3 binding, and you get elevated levels of the YAP and TAZ components, which are transcription factors that go into the nucleus. We actually discovered was when we went back to look at the other mutation, L205R, there's a subtle enhancement of YAP-TAZ signaling in these cells and no effective ERK signaling. Now remember, these mutations have the same residue, which is an arginine. They're 9 amino acids apart in the structure of the protein, as indicated here. So hipposignaling controls organ size, and MAP kinase signaling promotes adrenal hyperplasia. So here's the two mutations put on the structure. So this was pretty surprising to us for years of looking at signaling, that the downstream effects from these mutant kinases are very different. We can actually see that in the phosphoproteomes, which I'm not going to show you here. But what I am going to show you, I think, is the difference in locations in clinical samples. So you can actually compare W196R to L205R. And L205R is much more distributed regionally within the cell. It's all the way through the cell, and it's found in the nucleus, where it can have, that's maybe why it has a more common and profound effect, because it can certainly affect transcription. And I'll talk a little bit more about this later, because we have some interesting new data with this. So this is where we were about, I think in August, when we completed this paper, that we'd identified these two mutations. We had a mouse model. We had hallmarks of Cushing syndrome. And what we were really interested in was that these mutations, separated by 9 residues, promote distinct pathological signaling outcomes. They're excluded from ACAP signaling islands. But when you looked at the intracellular location in clinical samples, L205R to W196, it was different. And that was something that we were really interested in. So with that in mind, I'll tell you what we've been doing since then. So one of the advantages of working where I do is that we can get samples, we can get tissue sections of Cushing's patients. We've been working very closely with our Department of Lab Medicine and Pathology. The chair of the department is a good friend and colleague, Jeff Baird. And so what we decided to do was to take these samples. We get 10 serial sections. If you take 3 of these sections, you can isolate the DNA, and then you can PCR up the exons through 6 through 8 of PKA to look for these mutations. And so here's just a sort of proof of principle example, the most common mutation. We find an allele of L205 and R205. Now we've got other samples, so we can then go back and look at the adenoma versus the adjacent tissue, and you can see the difference pattern similar to what I just showed you. The other thing that we can do with these samples is we can check the biochemistry to see if, in this case, TAZ signaling is elevated, and you can see that it actually is here. This is actually in some ways more dramatic than the tissue samples I showed, and the cell samples that I showed. The other thing that we can, we were hearing about this a few minutes ago, take advantage of AlphaFold to look at what happens to the structure, because there's a lot of crystal structures of the catalytic subunit pKa. So here's L2O5R. If you look closely, the whole molecule just tilts and twists a little bit when you have this W196R. So we've been going on and we've been screening as many samples as we can get. And what we've discovered is a new mutation. At position 196, rather than an arginine, it's a glycine. And we've got multiple samples of this, which is shown here. In fact, at first, we didn't really believe this. This is now the new mutation that we've identified, which is tryptophan 196 to glycine. So you've gone from a bulky tryptophan to an arginine down to glycine, which is no side chain, basically. We've done deep sequencing on these patients to confirm that we have these mutations. You can see this here. We've compared the L2O5R to these two, in this case, just two. We've actually got, I think it's 4 of these patients now with the W196G, which is surprising because these are from a single cohort. And I was talking earlier about this. Because of the rules of confidentiality within the United States, we don't know if these patients are related, but we think they might be, which changes how these mutations occur. I can't confirm that because we just have legal sort of limits to be able to determine what's going on, but we're very excited about this mutation. And what I'm going to do is just take the last few minutes to tell you what we now know about this mutation, because it's a little bit different. So again, we've looked at tissue sections to see what the staining looks like. It looks somewhat similar to W196R, but it has a slightly more restricted subcellular distribution, and I'll explain that to you in a minute. There's enhanced cortisol production in these cells, not as dramatic as W196R, but still significantly enhanced and slightly above L2O5R. This is done, obviously, expression in cells. We're now making CRISPR-Cas alleles of these. But the surprising result when you compare these is that the type 1 regulatory subunit is significantly elevated in W196G than compared to L2O5R or compared to wild type. And this sort of elevation of the type 1 regulatory subunit is a fairly well-known pathological phenomena. And we normally look at the type 2 regulatory subunits, but we've now begun to look at both of these regulatory subunits. They're very similar, but they have some significant differences. So this is the autoinhibitory domain, or the pseudosubstrate site in R1. It's called the autoinhibitory domain in R2. These are fairly similar. They look like substrates for pKa. In one case, it is a serine that's the substrate. The other case, it's an alanine. And just downstream of these are this area here that's somewhat divergent. The main difference really is the presence of a proline in R2 that's not present in R1. So we can look at these, and this is the wild type situation. And again, using AlphaFold, we can predict changes in the structure based on that glycine mutation. And this work we've done with Safal Shariska and Nitan Kanan, where we've actually begun to do molecular simulations of these, which I hope will appear. Here we go. So in yellow, we have the type 1 regulatory subunit bound to the catalytic subunit in cyan, type 2 regulatory subunit bound to the catalytic subunit in cyan. They kind of wobble about for a while, but basically once they've stopped doing that, as this one has, you can see that first of all, there's more protein-protein interaction with R1 and this mutation than R2. There's more interaction at this area up here, and also in this area, if I can highlight this, you can see that there's additional interactions between this phosphothreonine 197 on the catalytic subunit and these two residues in R1 that is not present in R2 because you've got an imino bond, a proline bond there. So you've broken that interaction. Now, what this actually means is that this W196G mutation retains molecular interactions with the type 1 regulatory subunit. That gives it the capacity to be brought back into ACAP signaling animals. This is a different view of kind of what I've talked about before. And we'll see here, we've now been able to go back to use this super resolution live cell imaging with photoactivation to confirm if what I'm saying is correct, that the G mutant is retained within type 1 pKa signaling, ACAP signaling islands versus type 2. So this is the control here, wild type bound with the type 2 regulatory subunit. This is W196G that is not really retained very well within these complexes. You can see here, this is just the quantification of the data that compares in green W196G to W196R. They're fairly similar. Now we'll flip, this is an R type 1 selective ACAP called SMACAP, and the yellow staining is the type 1 regulatory subunit. And it's retained within the complex. Here's the mutant, and it stays within the complex as well. So there's a subtle difference, but a significant difference here, that the type 1 regulatory subunit can now hold on to this cushion. And you can see this here better when we've just looked at all the different cells. We did about 50 cells here. You can see that this is no statistical difference with the W196G to the wild type, but there is with the W196R. And again, when we do biochemistry and co-immunoprecipitation, you can see that the type 1 regulatory subunits, the G mutants, actually do bind the regulatory subunit, but the L205R absolutely doesn't. And again, we can look at ACAP, so recruitment into ACAP signaling islands. This ACAP called ACAP220 is actually a dual function anchoring protein. You can bind type 1 or type 2. It binds W196G and W196R, but not L205R. ACAP79 only binds type 2 regulatory subunits, so they're not found in the complex. And then finally, we went back to look at a couple of other mouse models that we have. So this is the heterozygous mutant I talked about earlier. And we do see the type 1 regulatory subunit elevated here, which is encouraging. This is an ACAP220 knockout mouse where we see the same phenomena. So what we're intrigued about is whether or not this has any adrenal effects. And we've not been able to see any differences in global cortisol production, but we've not really looked at this in any great detail at the moment. And then finally, what's interesting is we've been able to take cell lines and using siRNA, we can either remove all the regulatory type 1 subunit or all the regulatory type 2 subunit. And that's what these experiments are here. And we can measure cortisol release. So what you can see here is when you take out the type 1 regulatory subunit, cortisol levels go up. When you take out the type 2 regulatory subunit, they paradoxically go down. But this is actually because we think there's more type 1 regulatory subunit being produced. So enhanced expression of R1 alpha may be a compensatory mechanism to restore anchoring in these cells. And a lot of pathological phenomena that involve pKa have elevated type 1 regulatory subunit. We've not really known what that was for before, but we're now beginning to get some hints as to what this might be doing. So with that, and I probably should put the lights on so you can figure out how to do that, I will conclude that these Cushing's mutants are excluded from ACAP signaling islands. Directing Cushing mutants to ACAPs restores normal cortisol production. That potentially, if you could restore the defect, probably by CRISPR-Cas, would be an interesting experiment to see how we can do that. See if we can think about gene therapy. An adrenal expression of W196R promotes a Cushing's phenotype in mice. As I said, these two mutations have different downstream signaling effects. So just these subtle changes in the structure of the catalytic subunit have profound effects downstream. And then this new mutant is detected in about 20% of the patient samples we've tested so far. We've not tested a lot, and they've all been from one region, which is the Pacific Northwest in the US. So we don't know how general this is or what this means, but we're very excited to follow up because clearly it has different biochemical and cellular properties. So with that, I will conclude and give credit where credit is due. This work was almost all done at the University of Washington, mainly by a very talented postdoc, my lab, Mitch Omar, with some help from Tyler Lakey, and there's a few other people working on this. Antoine and Anne-Marie provided us with cells, which were invaluable to the work that we've been doing. And now the simulation and the sort of structural analysis we're working with Safal and Kanan in the University of Georgia. So with that, I will stop and thank you for your attention.